Hello, you guys. Today we're going to be doing chapter 1.2 of IB biology. So this chapter is slightly shorter than 1.1 and it's a bit smoother. So let's begin. So firstly, we're going to be talking about prokaryotes. So what is a prokaryote? Basically, a prokaryote is an organism which lacks a nucleus. And it's really, really small. It's actually between 1 and 10 micrometers. So now what makes a prokaryote a prokaryote? So what are its features basically? So firstly, you have the cytoplasm. So basically what the cytoplasm is that it's like the internal fluid of the cell, and all of the chemical reactions of life take place inside of the cytoplasm. And because the prokaryotes aren't compartmentalized, then all processes actually occur in the cytoplasm. Here it's written R, but they're actually not compartmentalized. And the nucleus. So basically, the nucleus is a region of the cytoplasm where the DNA is located. And you have to keep in mind that the DNA in the prokaryote is actually free DNA. And basically, plasmids. So plasmids are autonomous circular DNA molecules that have been transferred between bacteria. And basically, what they do is that they help adapt to changes in the environment. Ribosomes are complexes of RNA and protein that are responsible for protein synthesis. And basically, the DNA is not wrapped around the proteins. And now you have to remember this very, very well, is that the prokaryotic ribosomes have 70S. This is very important because we're going to be comparing prokaryotes to eukaryotes, which have 80S ribosomes. This is very important because it's really, really asked about when you need to compare eukaryotes and prokaryotes in both paper 2 and in paper 1 also, because usually they give you some sort of table and they're usually in it. So 70S and 80S are really easy to remember and they're really, really vital. And the cell membrane, so basically the cell membrane is a semi-permeable and selective barrier surrounding the cell. And the cell wall, basically what the cell wall is, it's like a rigid outer covering, and it's made out of the material peptidoglycan, and it protects the cell and maintains its shape, of course. And now we have the slime capsule. Now, of course, you have the image next to you, which is from BioNinja. So whenever I'm just explaining what each one is, you can directly just look at it in the image. You can refer to it in the image. So basically, the slime capsule is a thick polysaccharide layer for protection against desiccation and phagocytosis. Now you have to keep in mind that well, you should probably know what polysaccharides are, but basically you have monosaccharides, then you have disaccharides, and you have polysaccharides. So you have to make sure that you know these. But you're gonna learn more about these in detail in chapter 2 in the beginning. But of course, just know them. And you have a flagella. So basically, a flagella is a long, slender production containing a motor protein that enables movement. So basically, it's in charge of movement. And as well as, oh, flagella is actually not shown in this image, but flagella actually, they do quite, oh, these are the flagella. I think maybe because the image is not complete, but these are actually the flagella. These two are the flagella. And then you have the pili. So basically, the pili, they look like hair. So they're hair-like structures, and they basically enable adherences to surfaces or mediate bacterial conjunction. So basically what they do is that they allow bacterial cells to attach to each other and exchange DNA. This is what the pili does. So this is the pili. You can actually see they look like hair. So because you are actually required to draw a prokaryotic solid, prokaryotic cell, it's actually one of the objectives. So we're going to be discussing that later, but this is very important that you have to know how to annotate, because once they ask you to draw, it's very, very important that firstly, when you're given a certain like box, you have to make sure that your figure is within the box. You have to make sure that you annotate. You have to make sure that you label. You have to make sure that your lines are straight. And it's very, very, very preferable to actually use a pencil, and then over the pencil, you use a pen. It's very, very preferable. Now, we're going to be discussing binary fission. So basically, it's a form of asexual reproduction used by prokaryotic cells. Now, if you watched my previous video of the section 1.1, you would know that asexual reproduction does not involve any fertilization. Now, the process of binary fission. So firstly, and by the way, this is one of the only processes in all of IB biology where the processes are identical in many sources. So this is very important to know this word by word. So the circular DNA is copied in response to a replication signal. Then the two DNA loops attach to the membrane. After that, the membrane elongates and pinches off cytokinesis, forming two cells. So basically, this is a bit complicated because cytokinesis is actually in mitosis, which happens in the section 1.6. So if you don't know what it is, just know that it's part of mitosis, which is actually the division of cells, the replication of cells. But you don't actually have to know what mitosis is in detail until chapter 1.6. And at the end, the two daughter cells are generally are genetically identical. So basically, they're clones. So you can just look at this image, which actually really explains everything. Well, it's also from BioNinja. So you can see in the end that these two are identical cells. They're basically clones of one another, and you can actually see all of the process and how it's splitting into. Now we're going to be discussing eukaryotes. So this is actually the structure of a eukaryote. And basically, they're organisms which contain a nucleus. So just remember that the main difference between the prokaryotes and the eukaryotes is that the prokaryotes do not contain a nucleus and the eukaryotes contain a nucleus. So you have to know that the eukaryotes they're more evolved and they're more complex than prokaryotes, and they're actually bigger in size as well than the prokaryotes. They're also compartmentalized by membrane-bound structures by organelles. So basically, what it means is that it's split, and these organelles, each one of them, it performs a specific function. Now, why is it important, or why is it an advantage that there's compartmentalization in the eukaryotic cells? Firstly, it's better because it makes the cell more efficient. Basically, the enzymes and the substrates can be localized and more concentrated. So basically, if you don't know what enzymes are, basically enzymes are proteins, and what they do is that they make everything faster. Basically, they catalyze reactions. Enzymes are really important because humans cannot actually live without enzymes because they help us with digestion, they help us with processes, so we need enzymes. Again, we're going to learn about enzymes in chapter 2.4. So just know that it catalyzes reactions for now. This is if you're studying 1.2, but of course, if you're studying for the whole IB syllabus, whether you're SL or HL, you have to know what enzymes are, you have to know what mitosis are, all of these things, you have to know them in complete detail. Anyway, to continue, basically you have localized conditions. So basically the pH and the other factors can be kept in optimal levels. So basically what I mean is that the optimal pH level for one process is in one part of the cell. So like, for example, let's think of us human beings. So basically us human beings, what are we? We are eukaryotes. So basically, let's say our stomach has a specific pH because our stomach needs acid because it uses acid to actually digest. Let's say our mouth, we have salivary amylase in our mouth, which is another enzyme. But please, if you're only studying 1.2, don't focus on these details. I'm trying to explain. Or maybe you know this before pre-IB, because of course IB, you always have to have prerequisite knowledge. Anyways, in your mouth, there's a different optimal pH because there's different enzymes inside your mouth. So always keep that in mind. And we also have to know toxic, basically, there's toxic and damaging substances that can be isolated. Basically, if one organelle has a toxic or damaged substance, it means that it does not spread fast to the other organelles. So basically, you can have a digestive enzyme, and that enzyme can actually digest a leaf cell itself. And it's stored in the membrane sacs. So basically, instead of digesting the whole leaf, it's stored in a certain membrane sac. So I don't know if you understood, but basically these certain toxic, substances can be isolated once the cell is compartmentalized. Basically, they can be isolated in one single organelle. And the number and locations of organelles can be changed. So it basically depends on these cell requirements. So now we're going to be talking that we have to keep in mind that eukaryotes can be divided into 4 distinct kingdoms, but I'm actually not going to get into that because it's actually not in the IB syllabus for chapter 1.2 solely, but we will be discussing this in chapter 5. So keep in mind that you have to actually know these, but not for now. You actually have to know them in chapter 5. So just know that eukaryotes can be divided into distinct kingdoms such as Animalia, Plantae, but for now, just know that. Now, for the organelles, this is really, really important, and they're very, very asked about distinguishing between them. And in the MCQ questions, these are so important in the paper 1 questions. So, firstly, What are organelles? So we actually did go over organelles when we were talking about emergent properties in section 1.1, but to actually know the definition of an organelle, because in paper 2 it's very, very important. When you have a paper 2 question, you have to define, you have to explain, you have to, and of course, you have to pay attention to the command terms. And if you want to know more about the command terms, please join my classroom on Google Classroom. Now, an organelle is a a specialized structure within a cell that forms a specific function. Now we have to discuss universal organelles, meaning that they are both in eukaryotic and prokaryotic cells. And you have to always keep in mind that you're always giving these different types of tables throughout the years. They always give it in the IB exams, different tables with different organelles, and you have to figure out which one is suitable for the eukaryotes and the prokaryotes. So firstly, ribosomes. Ribosomes are two subunits of RNA and protein. So like I said, in eukaryotes, they are 80S, and keep in mind that the ribosomes in eukaryotes are larger than the ribosomes in prokaryotes, which are 70S in prokaryotes. And they do not have a membrane as part of the structure. This is very important to keep in mind that ribosomes do not have a membrane. And they occur within the mitochondria and the chloroplast. So basically, the mitochondria and chloroplast contain the ribosomes. And now you have to know the function as well of the ribosome. So what it does. So basically, it's the site of polypeptide synthesis. So basically, it synthesizes protein, because we do know that we have protein, then we have polypeptides, and then we have amino acids. We're going to learn about that again more in detail later, but just keep this in mind. Basically synthesizes proteins to function in cytoplasm within the cell, such as enzymes. Again, enzymes are proteins. We're going to learn about this more in detail. Just keep this in mind. It's very, very important. And the cytoskeleton. So basically, the cytoskeleton, it's not talked about in many IB resources, but we have to know this, because the thing about the IB biology especially is that you have to make sure that you go over all your resources, because again, it's a very, very, very wide curriculum. So the cytoskeleton is a filamentous scaffolding with the cytoplasm. So basically, it protects it. So basically, what it does is that it provides internal structure and mediates the intracellular support. So basically, it provides a structure which supports the cell, and it's less developed in prokaryotes. Again, the cytoskeleton is in both eukaryotes and prokaryotes. Again, these are universal organelles. Please keep that in mind. It's very important. And then we're going to be discussing the plasma membrane. So basically, it's a phospholipid bilayer. Again, this is the definition, but of course, we're going to actually learn about this in the next chapter, 1.3. It's a phospholipid bilayer with proteins, and it's a vital structure. So basically, the cell cannot live without the plasma membrane. And what it does is that it's a semi-permeable and selective barrier surrounding the cell. So basically, it's in charge of what goes inside the cell and outside the cell, basically. And it retains the fluid cytosol, which is basically cytoplasm. And it is where the cell is identified by surrounding cells. So basically, when you see a plasma membrane, you know that this is one cell, and you see a plasma membrane next to it, it contains another cell. So basically, this is how you differentiate. I don't know if I understood, but basically, the cell is covered by the plasma membrane, and this is how we can define one cell as the cell being itself. Now we're going to be talking about the eukaryote organelles. So basically, organelles that are solely and only keep in mind in eukaryotes. And again, I show the pictures because this is very, very important, because again, you are shown images in the paper 1 questions, and you are supposed to identify. I'm going to check, but for example, this is very important. You see, I didn't go over this. So basically, you have the free ribosomes and you have the bound ribosomes. So basically, the free ribosomes, as you can see, they're here. But the bound ribosomes, they are attached. They're here. So just keep this in mind. So these are like the free ribosomes all over. And these are the ones that are bound ribosomes. So the eukaryotic organelles. So firstly, you have the nucleus. Again, why I said it includes both animals and plant cells, because like I previously mentioned in the section of 1.1, again, if you didn't watch, it's very vital to do so, because again, it's an accumulation. You get to know more and more. And actually, once you learned all of the IB syllabus and you're reviewing for your official exams or your finals or your mocks, everything will start to build up and make much more sense, because everything will interconnect. So the more you build on your knowledge, the more you study the chapter thoroughly, the easier it will become. Now, a nucleus. Oh yeah, like I was saying, plant and animal cells, they differ, but they are both eukaryotes, because like I said, there are 4 kingdoms in the eukaryotes, and Animalia and Plantae are included, and that's why the plant and animal cells do have differences, but they are both eukaryotic, and they both do have similar organelles, which we are going to be talking about now. So first you have the nucleus. So it's a double membrane structure with pores, and it contains an inner structure called the nucleolus. So basically what the nucleolus is that it's a tiny rounded darkly stained body, and the nucleus itself, it has pores and holes that are present in the membrane, and it contains genetic information which are in the form of both chromosomes and chromatin. And basically chromosomes are the condensed form of chromatin. Now you actually do have to know what chromosomes and chromatin are. We're going to learn about this in chapter 3, which is genetics, but just know that chromosomes basically means DNA. And this is a picture of the nucleus. It's very, very important to know how to identify it. And what the nucleus does is that it stores genetic material in the which is DNA as chromatin, and it's the site of the ribosome assemblies. This is very important because you are always asked to define them and for their functions as well. So the endoplasmic reticulum. So it's a membrane network that may be bare. So basically you have two types of endoplasmic reticulums. You have the smooth endoplasmic reticulum and you have the rough endoplasmic reticulum. Now what's the difference? The difference is that the rough endoplasmic reticulum is studded with ribosomes, and that's why it's much easier to identify. And a hint when you're trying to identify them in the MCQ questions, or even in the paper 1 and paper 2 and paper 3 questions, you're also asked to identify certain structures. They give you an arrow, they point you to that structure, and you have to be able to identify them. So now the smooth endoplasmic reticulum, besides the fact that remember, keep in mind, it's not studded with ribosomes, it actually produces lipids. Whereas the rough endoplasmic reticulum, it helps modify and transport lipids, and it's also closer to the nucleus. Remember, whenever you're trying to identify the endoplasmic reticulum, make sure to remember two things. Firstly, that it's close to the nucleus. Second, if you want to distinguish if it's rough or smooth, the rough one has ribosomes attached to it, whereas the smooth does not. Now we're going to be talking about the Golgi apparatus. So basically the Golgi apparatus is found in all cells, but it is prominent in metabolically active cells. Basically what it is, it's an assembly of folded vesicles and membranes located near the cell membrane. So it's located near the cell membrane and it's basically folded. And that's why you'll see that the Golgi apparatus always has a folded membrane, especially in your biology books, you'll see that. And now it has many functions. So basically, it's involved in storing and sorting and modifying and exporting secretory products. So basically, one side of the Golgi is near the rough endoplasmic reticulum, and it receives proteins and other materials and then packages them. And then in membranous sacs, keep in mind, these membranous sacs are called vesicles. The vesicles exit out of the other side of the Golgi, and always write Golgi apparatus, it's better, but I'm just writing Golgi to make things faster. And the vesicles can either be transported within the cell or they can be shipped out of the cell. And keep in mind, the vesicles are membrane sacs, and the cells produce a lot of product for other areas of the body and have a lot of Golgi. Keep that in mind. Now the mitochondria. So basically, it's a double membrane structure. Keep in mind, it's double membrane structure. This is very, very important. And the outer membrane is a smooth boundary, but the inner membrane is folded to form cristae. So what is cristae? Because when I was studying the chapter myself, I was like, what is that? But actually cristae is, it increases the surface area and it speeds up chemical reactions. So what the function of the mitochondria is, it's a site of aerobic respiration. Now we're going to be learning, like I said in the previous video of section 1.1, that we're going to be learning about respiration in chapter 2.8, but you have to know that aerobic respiration basically requires oxygen, and one of its product is ATP. ATP is basically adenine triphosphate, which provides energy for the cell, because it actually, for example, many functions take place due to ATP because we need it. Anyways, a peroxime is a membranous sac containing a variety of catabolic enzymes. Basically what it does, because the minute we see enzymes and catabolic, we directly know that it catalyzes and breaks down toxic and toxic substances and other metabolites. And you have to keep in mind, I didn't talk about the structure of the membrane, but the structure of the mitochondria, but the structure of the mitochondria is actually the easiest to identify. It just looks like it's always the same in all the pictures. It just looks like that's really easy to identify. Now, the centrosome. So basically, it's a microtube organizing center, and it's located near the nucleus. Now, it's not really much talked about. It hasn't really been asked to identify, but just know what it is. And the function is that it radiates microtubules to form spindle fibers and to contribute to division. So basically, you're going to actually be talking about that deeply or in depth in chapter 1.6. So just keep that in mind. Know what the centrosome is. That way you can actually build on your knowledge. So basically, it also helps to the movement of chromosomes during cell division, like I previously said. But for now, just know that it's a microtubule organizing center, because you don't have to know this for chapter 1.2, but for chapter 1.6, you definitely have to know this. Now, plant cells. So basically, these organelles that we're going to be discussing are only and solely in plant cells, because again, you are required to know the difference between animals and plant cells. Basically, the chloroplast, it's basically a double membrane structure. Keep in mind it's double membrane because you're going to be asked which organelle has a double membrane, so always keep that in mind. With internal stacks of membranous discs. So these membranous discs are called thylakoids, and basically are going to be learning more about this very in depth if you are HL. Basically, you're going to be learning about this in chapter 8. But for the SL students, you're going to be learning about the process of photosynthesis in chapter 2.9, but not in depth. It's going to be very broad, very light. So basically chloroplasts. So what chloroplasts are, they are plasmids, and they're found in many plant cells, but never in animals, because basically chloroplasts, they're organelles that contain the pigment chlorophyll, and they are responsible for the process of photosynthesis, and it cannot take place without the chloroplast. It's a green pigment, basically, but just know for now the definition, which it's a double membrane structure, and it has internal stacks of membranous discs. And keep in mind that you have to know the image, how it looks like, and just make sure that you don't get confused between it and the mitochondria, because as you can see here, their shapes are slightly similar, but what they contain is different, because the chloroplast contains chlorophyll. So keep in mind when you're looking at sacs, it's sacs of chlorophyll. Now, the function is the site of photosynthesis, like I mentioned. Now the vacuole. So basically, it's a fluid-filled internal cavity surrounded by a membrane, and it has a single membrane. So keep that in mind. It's very important. Now the function of a vacuole. So basically, it maintains a hydrostatic pressure. So basically, what the hydrostatic pressure is that it just keeps the all of the water inside of the cell in order for it not to burst. And basically in vacuoles, we did mention this in the previous video. So basically in plants, the vacuoles are permanent and large, whereas in animals, the vacuoles are temporary and small. That's why we put it in plant cells, because in animal cells, the vacuoles are completely different. And now plant cells, now this is very, cell wall. So this is very important in plant cells, because basically they're external covering made out of cellulose. So basically the cell wall is not an organelle. So please keep that in mind. It's mentioned in many sources that it's not an organelle. It's permeable, strong, and hard to digest, because us humans, for example, we do not have the enzymes to digest cellulose. We are not able to digest cellulose. And the function is for it to provide support and mechanical strength. And basically, it prevents the plants from having excess water uptake. That way, the plant will actually burst. So it provides structure, basically. And this is a typical plant cell. You can see the chloroplast, and you can see inside it. This is what I was talking about here. And you can also see the cell wall, which is basically made out of cellulose. This is it. And this is what I was talking about. The Golgi apparatus is always folded in this shape. So keep all of this stuff in mind. And here you can see the difference between the smooth and the rough endoplasmic reticulum. These are the ribosomes. They're studded with ribosomes, whereas here there isn't any ribosomes. So keep this in mind. Now for the animal cells. So basically there's lysosomes, which again we mentioned in the previous video. They're membranous sacs filled with hydrolytic enzymes. They're formed of the Golgi vesicles. Very, very, very important. They have a high concentration of enzymes, which causes them to have a heavy stain. So basically, once there's actually a heavy stain, you're able to determine them in the micrograph that this is the lysosome. So basically, what does the lysosome do? So basically, the lysosomes, they break down macromolecules, and there's kind of a debate that lysosomes are present in plant cells, but for now I always say that they're only present in animal cells. They are have digestive enzymes that basically they contain digestive enzymes, and what these enzymes do is that they ingest food in vesicles. They get rid of unwanted and damaged cells, and that they can actually break down the cell itself. Now, this is a typical animal cell. So it contains, for example, the cytosol basically is a cytoplasm. So keep that in mind. The cytoskeleton, which I previously mentioned, the mitochondria, and this is the Golgi body that's folded, like I said. This is the plasma membrane, the rough endoplasmic reticulum, which contains the ribosomes everywhere, and this has no ribosomes. The nucleus, which contains the nucleolus. So basically, these are the ribosomes. They're free ribosomes. They're floating. They're not attached to anything. So yeah, and this is the lysosome here. This is it. And now for the electron microscope. So firstly, the electron microscope use electron beams focused by electron magnets to magnify and resolve microscopic specimens. So like I said, I mentioned the definition of resolution in my video in section 1.1, but basically resolution is when you can be able to distinguish between two objects, and if these two objects are not able are not being able, like you're not being able to tell them apart, it means that they are one object. And the advantages of electron microscopes to light microscopes is that they have a higher range of magnification, meaning that they can actually see smaller structure because of the electromagnets, and they have higher resolution, hence can provide clearer and more detailed images. And it's very, very important, because you're always asked about what's the advantage between electron microscopes and light microscopes. It's always that electron microscopes, they're able to have a higher resolution. It's not they're able, they actually have a higher resolution, which gives them the ability to see these structures more clearly and more detailedly. This is why. So always keep that in mind, the higher resolution. So whenever you think about light versus electron microscope, always remember the higher resolution part. Now, this is a quick revision for the important tables you should know. So you should know prokaryotes versus eukaryotes. So basically, prokaryotes, they have naked DNA, like I previously mentioned, whereas eukaryotes, they have DNA that's bound to proteins. Again, these are both from the source Bar Ninja. The prokaryotes, their DNA is circular, whereas the eukaryotes, their DNA is linear. The DNA usually has no introns in prokaryotes, but in eukaryotes it has introns. In organelles, the prokaryotes have no nucleus, which they are in fact known for. This is what makes prokaryotes prokaryotes. They are organelles that do not contain a nucleus. Whereas the eukaryote has a nucleus. Again, they're not the organelles and prokaryotes are not membrane bound, and like I previously mentioned, they are not compartmentalized. Whereas the eukaryotes, they are membrane bound, they are compartmentalized. And again, the most important is the 70S ribosomes for prokaryotes and the 80S ribosomes for eukaryotes. And the binary fission take is how prokaryotes reproduce, whereas eukaryotes reproduce through mitosis and meiosis, which we're going to be discussing. We're going to discuss mitosis in chapter 1.6, and in meiosis we're going to be talking about in chapter 3. And then prokaryotes have single chromosome, which makes them haploid, and eukaryotes have chromosomes paired. Again, please don't get to these details for now. And the average size is 1 to 5 micrometers, whereas for eukaryotes it's 10 to 100 micrometers. So basically, eukaryotes are significantly larger, and their organelles are significantly larger. So now for plants versus animals. So basically, plants, they have plastids, such as the chloroplast, like I previously mentioned, whereas animals, they do not have plastids. Again, plastids are never in animals. Plants have a cell wall made out of cellulose. Animals do not have a cell wall. The plants, they have large contractile vacuoles, whereas the animals, they have small and temporary vacuoles, if they have, because in many cases they do not. And then plants have a plasmodesmata, whereas animals do not. Plants do not have centrioles, but animals do have centrioles, and these centrioles are inside the centrosomes. Lastly, plants do not have cholesterol in cell membranes, whereas animals have cholesterol in cell membranes. And plants, okay, this is a bit not for now, but plants, they do store excess glucose as starch, because they actually do produce food as starch through the process of photosynthesis. And animals, they store excess glucose as glycogen. Again, glycogen is all only in animals. And plant cells generally have a regular shape, whereas animal cells have a amorphous shape. Thank you so much for watching, guys. This is it. If you want any practice on paper 2 questions, please comment down below, because I actually do have paper 2 questions. And please join my Google Classroom. The link is down below if you want more notes, more resources, my personal notes, all of that. Thank you so much, guys, for watching. Bye bye, and enjoy your day.